Good morning, everyone. Welcome to Mediquaries. Today, we are going to discuss an important vascular carrier systems in the field of novel drug delivery systems, that is, ethosomes. Ethosomes are the specialized liquid vesicle, particularly designed to improve the transdermal and the topical delivery of the drugs here. So, the major distinguishing feature of ethosomes is their relatively high concentration of ethanol. So, ethanol mainly imparts the flexibility to the vesicle membrane and also modifies the lipid organization of the stratum corneum here. So, in this lecture, we will cover the definition, structure, composition, mechanism of penetration, preparation methods, formulation, variabilities, characterization, advantages, limitations, applications, and comparison with other vascular systems. By the end of this lecture, you should be able to explain not only what ethosomes are but also why ethanol makes them particularly effective for the transdermal drug delivery here. Okay, so after completing this lecture, we should be able to define the ethosomes and explain their structural characteristics. So we will identify the important components of an ethosomal formations and understand the role played by each component here. So we will then discuss the mechanism by which ethosomes cross the stratum corneum. So another important objective is to understand the different preparation methods, particularly the cold method, which is widely discussed for the ethosomal formulations here. So we will also explain the important evaluation parameters such as vesicle size, PDI, zeta potential, entrapment efficiency, deformability, drag release, and the skin permeations. So finally, we will compare the ethosomes with the conventional liposomes, transfersomes, and transethosomes, right? So why transdermal drug delivery? So before understanding the ethosomes, we need to understand the basic challenge associated with the transdermal drug delivery here. So the skin is not simply a protective covering, it is a highly organized biological barrier. So for systemic transdermal delivery, a drug must pass through the several skin layers before reaching the systemic circulation here. So the most important barrier is the stratum corneum. So it is composed primarily of the dead keratinized cells called the corneocytes embedded in an extracellular lipid matrix here. So this structure is often described using the brick and mortar model. Okay, so the corneocytes acts like a brick while the intracellular lipids behave like a mortar here. So this highly organized arrangement prevents many molecules from penetrating through the skin, right? So therefore, although the transdermal delivery has several advantages, achieving the adequate drug flux through intact skin is challenging here. So this is preciously where advanced vascular systems such as ethosomes become useful. Okay, so problems with the conventional transdermal delivery. So several physiochemical properties determine whether a drug can be successfully cross the skin or not. So the drug should generally possess a suitable molecular weight, adequate lipophilicity, sufficient aqueous solubility, and appropriate partitioning characteristics. So large molecules, highly hydrophilic molecules, and the molecules with the poor partitioning characteristics often show the inadequate skin permeation here. So furthermore, the stratum corneum creates a significant diffusion barrier. So the conventional creams, gels, and ointments may provide the local delivery, but achieving the deep penetration or systemic delivery can be difficult. So the chemical penetration enhances can be used, but excessive enhancement may produce the irritation or compromise the barrier. So therefore the researchers developed the physical and the carrier based approaches here. So one such approach is the development of the ultra deformable liquid vesicle including ethosomes. So let us see the definition of the ethosomes first. So ethosomes are the solid, soft, flexible, liquid-based vesicular carrier containing a phospholipid, a relatively high concentration of the ethanol and the water, designed primarily for enhanced delivery of the drug through the skin here. So the term ethosomes is derived from the presence of ethanol in the vascular system. So unlike the conventional liposomes, so ethosomes contains a suitable amount of ethanol here. So this ethanol has two important effects. So the first effect is it modifies the properties of the vascular membrane and increases the flexibility of the vesicle. So that's the first. And the second is the ethanol interacts with the lipids of the stratum corneum and increases their flexibility. So overall the ethanol increasing the flexibility of the vesicle itself and also the skin. So therefore the ethosomes are not simply a conventional liposomes with ethanol added. So their improved transdermal performance mainly results from the combined effects of the vesicle deformability, ethanol skin interaction and enhanced drug partitioning and transport here. Okay, so historical background. Ethosomal technology emerged from the research into improved transdermal drug delivery using an alcohol containing lipid vesicles here. The concept was particularly associated with the work of co-workers who described ethanol containing phospholipid vesicles capable of improving the dermal and the transdermal delivery here. So this was an important development because conventional liposomes generally have a limited ability to penetrate the intact skin deeply here. So the incorporation of the ethanol means what happening here, that there will be a changing in the physical properties of the vesicle and their interaction with the skin. So since then ethosomes have been investigated for delivery of the wide variety of the compounds including anti-inflammatory drugs, antifungal drugs, antiviral, hormones and cosmetic activities. Okay, so here the main advantages of this ethosomal technology is, here we are using the ethanol, thereby there will be increasing in the flexibility of the vesicle itself and also the drug penetration through the skin can be increased by using of this ethanol here. Okay, so that's the main difference. So here the rate of penetration is also more and deep compared to other liposomes. Okay, so let's see the basic structure. So the structurally an ethosomes mainly consists primarily of a lipid bilayer surrounding an aqueous internal environment here. So the major structural component is usually a phospholipid here. So the phospholipid molecules contains a hydrophilic head and a hydrophobic tails. We are all know about that. So in an aqueous environment, these molecules arrange themselves into a bilayer structures here. So the drug may be located within the aqueous core associated with the lipid bilayer or distributed between these regions depending on its physiochemical characteristics, right? So ethanol is incorporated into the system and modifies the membrane properties here. So because of the ethanol, the lipid bilayer becomes more flexible and less rigid compared with more conventional liposomal systems, right? So this flexibility is extremely important for the transdermal permeation. Okay, so let us see the composition of the ethosomes. So an ethosomal formulation generally contains four major components. The first one is the phospholipid, and second is the ethanol, third is water, and fourth is the drug or active pharmaceutical ingredient. So depending on the formulation, additional components such as cholesterol, glycols, antioxidants, preservatives or edge activators may be included. So the exact composition is not fixed here. Okay, so it depends on the drug, intended route, desired vesicle characteristics, stability requirements and the skin permeation target, right? So let us see the role of the phospholipid. So phospholipids from the structural framework of the ethosomal vesicles. So commonly phospholipids investigated includes the phosphatidyl okay, so phosphatidyl ethanolamine and related phospholipid materials are used. So the phospholipid concentration can significantly affect the vesicle size, membrane characteristics, entrapment efficiency and the stability here. So if the concentration is too low, the formation of the stable vesicle may be difficult here. So if it is extremely high, the formulation may becomes more viscous and vesicle aggregation or the size changes may occur. So therefore, the phospholipid concentration must be optimized experimentally here. Okay. So that's the main goal. Role of ethanol. So ethanol is the most characteristic component of the ethosomes. So its role is multifaceted. So the first, ethanol interacts with the phospholipid molecules and increases the fluidity of the vesicular membrane here. Second, it contributes to increase the vesicle deformation. Third, when the ethosomes contact the skin, so the ethanol can interact with the lipid domains of the stratum corneum and increases the lipid fluidity. So this can reduce the barrier resistance temporarily and facilitates the penetration. So however, the ethanol concentration must be carefully optimized here. So too little ethanol may not provide the sufficient flexibility or penetration enhancements. So too much ethanol may destabilize the vesicle, alter the membrane integrity and increases the evaporation or potentially causes the skin irritations also. So therefore the ethanol represents both the strength and one of the formulation challenges of the ethosomes here, right? And role of water. So water acts as in a continuous aqueous phase here. So it mainly hydrates the phospholipids and provides the environment required for the vesicle formation. So the water to ethanol ratio influences the vesicle characteristics here. So the changes in the hydration conditions can affect the vesicle size, lamellarity, drug entrapment and the stability. So therefore the water is not merely a diluent here. So it is an important formulation variable, right? And role of drug. So the drug can be incorporated into the ethosomes depending on its physiochemical properties. So a hydrophilic drug may preferentially occupy the aqueous component. A lipophilic drug may associate the predominantly with the phospholipid bilayer. So amphiphilic drugs may distribute between both the regions. So the drug's molecular weight, log P, aqueous solubility, ionizing state and membrane affinity all influences its incorporation and the release. So therefore the formulation strategy must be selected based on the characteristics of the drug rather than simply applying one standard composition to every molecule here. Okay. And optimal components. So additional excipients may be incorporated when required here, like cholesterol. Cholesterol can be influences the membrane rigidity, permeability and the stability here. So however, increasing the membrane rigidity excessively may reduce the deformability that is important for the ethosomal preparation and the penetrations here. So the propylene glycol or other glycos may be used in certain formulations. So the antioxidants may be incorporated when phospholipid oxidation is a concern here. So the preservatives may be required depending on the formulation and the intended use. So for specialized systems, surfactants or the edge activators may be included, leading towards the systems such as ethosomes. So why ethosomes are flexible? So one of the most important concept in the ethosomes is the deformability. So a conventional rigid vesicle encounters a difficulty when attempting to cross the narrow pathways within the skin. So an ethosome, however, can undergo the deformation. So its membrane can temporarily change the shape while maintaining the vascular integrity here. So this allows the vesicle to move through the narrow intercellular pathways more efficiently here. So the high ethanol content contributes significantly to this flexibility by modifying the packing of the phospholipid molecules. So therefore, the ethosomal deformability is closely associated with their enhanced skin penetration, right? Skin structure. So to understand the ethosomal penetration, let us briefly review the skin anatomy here. So the major layers includes the epidermis, dermis and the subcutaneous tissues. So the outermost portion of the epidermis is the stratum corneum. So the stratum corneum is the principal barrier to the transdermal drug delivery transport here. So below it is variable epidermis followed by the dermis. So depending on the therapeutic objectives, a formulation may be designed for superficial deposition, deeper dermal delivery or systemic absorption. So ethosomes are particularly interesting because they can improve the movement across the stratum corneum and increases the drug deposition in the deeper skin layers here. Okay, so the deeper drug delivery can be highly achieved by use of ethanol. Okay, so that's why these are the more effective type for drug delivery systems. So mechanism of the ethosomal penetration. So now we reach the most important section, the mechanism of actions of the ethosomes here. So the enhanced delivery can be understood through several complementary mechanisms. So first one is the ethanol increases the fluidity of the ethosomal membrane. Second, the ethanol interacts with the stratum corneum lipids and increases their fluidity. Third, the flexible vesicles can deform and pass through the narrow intracellular pathways here. Fourth, the concentration gradient generated by the formulation promotes the drug movements into the skin. So rather than relying on only one mechanism, ethosomal delivery involves a combination of the ethanol induced skin modification here. So the vesicle flexibility and enhanced drug transport. Okay, so stepwise mechanism. So let us describe the process step by step here. So initially the ethosomal formulation is applied to the skin. So as the ethanol interacts with the stratum corneum, the ordered lipid structure becomes more fluid here. So at the same time, the ethanol containing the vesicles possesses the increased flexibility here. So the vesicles approaches and interacts with the altered skin barrier. So because of their deformation, they can pass through the narrow pathways. So as the vesicles move deeper, the drug is released or deposited within the skin here. Is it clear? So depending on the drug and the formulation, the drug may be subsequently entering the dermal circulations and eventually the systemic circulation here. So this explains why ethosomes can provide the better transdermal delivery than the conventional liposomal system. Routes through skin. So there are mainly three conceptual pathways for the drug movements across the skin. The intracellular pathway involves the movement between the corneocytes. The transcellular pathway involves the movement through the corneocytes, and the appendicial pathway involves the structures such as hair follicles and the sweat glands. So for many conventional molecules, the intracellular lipid pathway represents a major barrier here. So the flexible vascular systems may exploit these pathways more efficiently and effectively also. So the precise dominant pathway can vary according to the formulation, composition, drug properties, skin conditions and the experimental methodology here. Thermodynamic activity. Another important concept is the thermodynamic activity. So the driving force of the drug movement across the skin depends on the chemical properties of the thermodynamic activity of the drug here. So ethanol can influence the drug solubility and the partitioning. So during applications, evaporation of the volatile components may further alter the local composition, and the potentiality increases the activity of the drug here. So this can contribute to the stronger driving force for drug penetration into the skin. So therefore, the ethosomal penetration is not explained only by the vesicle deformation. So the physiochemical environment surrounding the drug is also an important factor here, right? Ethosomes skin penetration. So when ethosomes reaches the stratum corneum, several interactions occur here. So ethanol can penetrates into the lipid domains of the stratum corneum. This can disturb the highly ordered lipid arrangement. So the barrier becomes temporarily more fluid here. So meanwhile, the ethosomal vesicle itself remains sufficiently flexible to deform here. So this creates a favorable situation for drug transport. So it is important to emphasize that the exact molecular mechanism can depends on the formulation composition and the experimental conditions here. So therefore, the mechanism should be understood as a combination of the ethanol skin interactions and the vesicle mediated transport rather than as a single universal pathway. Preparation general principle. So ethosomes can be prepared using different techniques. So the objective is to obtain the vesicles with the appropriate size, uniformity, deformability, drug entrapment, and the stability. So the most commonly discussed approaches includes the cold method, hot method, and various mechanical dispersion approaches. So among these, the cold method is particularly useful because it avoids prolonged exposure of the formation of the high temperatures here. Let's now understand the cold method in detail. So in the cold method, the phospholipids and the drug are dissolved or dispersed in the ethanol. So the mixture is maintained under controlled stirring. So the aqueous phase is then added slowly to the ethanolic lipid phase. So this controlled addition promotes the formation of the vesicular structures. So the dispersion may be subsequently be subjected to sonication or extrusion if further size reduction and the homogenization are required here. So the final formulation is stored under appropriate conditions. The exact process parameters includes the mixing rate, temperature, addition rate, and composition must be optimized experimentally here. Okay. Cold method. So the process can be memorized as the phospholipid plus ethanol, drug incorporation, controlled hydration, vesicle formation, size reduction, characterization, and storage. So the key point is controlled mixing here. So the rapid addition of the water or the uncontrolled processing can produce the heterogeneous vesicles in place of homogeneous vesicles. So because of all these factors, mainly the rapid addition of water may contributes to the formation of the heterogeneous vesicles here. So therefore the process parameters are very important for the reproducibility here. Hot method. So in the hot method, the aqueous and the lipid phases are prepared separately using in a controlled temperature conditions. So the phospholipid containing phase is heated appropriately, and the aqueous phase is maintained at a comparable temperatures here. So the two phases are then mixed to produce the vesicular dispersion here. So after formation, the preparation is cooled and processed further if necessary. So the hot method can be useful for certain formulations, but heat sensitive drugs and excipients require the careful considerations here. Sonication and extrusion. So after preparation, the vesicles may be too large or heterogeneous in size. So therefore the secondary processing step here is the sonication or extrusion. So sonication uses acoustic energy to produce or to reduce the vesicle size here, and extrusion forces the vesicular dispersion through membranes with defined pore size. Okay, so the extrusion can improve the size uniformity and produce a narrow particle size distribution here. So however, the excessive mechanical energy can potentially affects the vesicle integrity here. So therefore the processing conditions must be optimized rather than maximized. Okay. Formulation variabilities. So ethosomal preparations, mainly the performance is strongly dependent on formulation variabilities. So important variabilities mainly includes the phospholipid concentration, ethanol concentration, drug concentration, cholesterol concentration when used. Okay. And aqueous phase composition, mixing conditions, temperature, processing time, and vesicular size. So all these variables can mainly influence the particle size, PDI, zeta potential, entrapment efficiency, deformability, drag release, skin permeation, and the stability of the final preparation here. Okay. So effect of the ethanol concentration. So let us see about this. So let us examine the ethanol concentration in the greater detail here. So increasing the ethanol generally mainly increases the membrane fluidity and can improve the deformability up to an optimum range here. So however, the excess ethanol may disturb the vesicular structure. So it can also increase the possibility of the skin irritations and the formulation instability here. So therefore the relationship between the ethanol concentration and the permeability is usually not a simply linear here. So in formulation development, an optimum ethanol concentration must be identified experimentally here. So this is a classic example of why the pharmaceutical formulation is an optimization problem rather than a simple ingredient selection exercise here. Effect of phospholipid concentration. So the phospholipid concentration mainly influence the amount of the lipid available for the vesicle formation here. So increasing the phospholipid concentration may increase the drug entrapment for certain drugs. So it can also alter the vesicle size and the membrane properties here. So however, the excess phospholipid concentration can increase the viscosity and may produce the larger or more heterogeneous vesicles. So therefore the optimum concentration depends on the drug and desired delivery characteristics, right? And drug entrapment. So drug entrapment efficiency describes the percentage of the drug incorporated into the vesicle systems relative to the total amount used during the preparation. Okay. Is it clear? Again, I'm repeating here. So the entrapment efficiency mainly describes the percentage of the drug incorporated into the vesicles compared to the total amount of the drug used in the preparation. Okay, so it can be calculated as the entrapment efficiency equals the amount of the entrapped drug divided by the total drug added multiplied by 100. So this method mainly used to separate the free drug from the vesicle associated drug is critical here. So the common approaches includes the centrifugation, ultracentrifugation, dialysis, or size exclusion techniques depending upon the formulation here. So the analytical method must be validated because inaccurate separation can produce the misleading entrapment values here. Vesicle size. So vesicle size is one of the fundamental characteristic parameter here. So it mainly influences the physical stability, skin interaction, drug release, and the potentially permeation here. So dynamic light scattering is commonly used for the nanoscale and the sub micro particle size measurements here. So the result should not be reported simply as one size value. Okay, so we should also examine the polymer dispersibility index, so which mainly provides the information regarding the size distribution here. So a lower PDI generally indicates a more uniform population. So although the acceptable value depends on the intended applications and the measurement conditions here. Okay, and next is the zeta potential. So zeta potential is the another important parameter. So it mainly represents the electrokinetic potentiality near the slipping plane of dispersed particles. So it mainly provides information related to the surface charges, okay, that is surface charges, and can help predict the electrostatic interactions between the vesicles here. So however, the absolute zeta potential values may contribute the electrostatic stabilization in some systems. So however, the ethosomal stability cannot be predicted from the zeta potential alone, because ethanol, phospholipid composition, steric effects, and other formulation factors also contribute here. So therefore, the zeta potential should be interpreted together with the particle size, PDI, morphology, and physical stability studies here. Morphology. So morphological evaluation helps confirm the vesicle formation and the structural characterization here. So the techniques such as transmission electron microscopy, scanning electron microscopy, cryo-TEM, and atomic force microscopy may be used depending on the research objectives here. So the microscopy can mainly provide the information about the vesicle shape, surface characteristics, aggregation, and the structural integrity here. So however, we must remember that the sample preparations can alter the vesicles. So therefore, the microscopic images should always be interpreted together with other characterization techniques here. Deformability. So deformability is particularly important for the ethosomes. So a deformability test evaluates the ability of the vesicles to pass through the membrane or a narrow pore under a defined pressure or the driving force here. So the amount of the vesicle material passing through the membrane and the resulting vesicle characteristics can be used to estimate the deformability here. So the higher deformability generally indicates the better ability to undergo the shape changes. However, this deformability alone does not automatically guarantee the superior skin penetration. So the entire formulation and the drug skin interaction must be considered here. In vitro drug release. So in vitro drug release studies determines how the drug is released from the ethosomal systems. So methods may include dialysis membrane method system or diffusion cells. So the release profile can be fitted to the mathematical models such as zero order, first order, Higuchi, or Kosmeyer-Pappas models where appropriate. Okay. So however, the mathematical fitting should not be used blindly here. So the selected model should be a mathematical justification and should be interpreted based on the experimental design. Ex vivo skin permeation. So ex vivo skin permeation studies are particularly important for the transdermal formulations here. So exercised animals or human skin may be used depending on the ethical approval and availability and the regulatory requirements. A Frange diffusion cell is commonly employed here. So the formulation is placed in the donor compartment while the receptor compartment contains an appropriate medium. So the samples are collected at predetermined intervals and the resulting data can provide the cumulative drug permeation, steady state flux, and permeability coefficients and the lag time here. So these parameters are critical for assessing the transdermal performances here. Frange diffusion cell. So in Frange diffusion cell, the skin is positioned between the donor and the receptor compartments. So observe the diagram here. So there are two compartments over there. That is the one is the donor compartment and the receptor compartment. So between those two compartments, we are attempting to fix the skin. Okay. So the receptor medium is maintained at a particularly controlled temperatures and the stirred continuously here. So the formulation is placed on the skin surface. So the drug molecules migrates to the skin and enters the receptor compartment here. So samples are withdrawn periodically and replaced with the fresh medium. Okay. So from the concentration versus time data, we are calculating the cumulative parameters, that is cumulative permeation and the steady state flux here. So this is one of the standard experimental approach for comparing the ethosomal formulations with the conventional formulations. Okay, so the main advantage of this method is here using or you are creating the in vivo environment itself in the in vitro by using the skin. Okay, so here what's the environment, what's the actual drug facing is we are highly creating here artificially, that is in vitro by using the Franz diffusion cell. So here it is a more effective method. And permeation parameters. So three important permeation parameters are commonly discussed here. The first is the steady state flux, usually expressed as amount per unit area per unit time. So the second is the permeability coefficient, so which is the mainly relates the flux to the concentration gradient. And third is the lag time, so which mainly represents the time required before steady state permeation is established. A successful lithosomal formulation may demonstrate increased flux and reduce the lag time. However, the interpretation should always be based on the statistically valid comparisons. Drug deposition in skin. So now not every transdermal formulation needed to be delivered the drug completely into the systemic circulation. So for dermatological theory, or so the objective may be to increase the drug concentration within the skin itself. So therefore the skin deposition studies are highly relevant here. So after the permeation experiment, the skin can be processed and the amount of the drug retained within different layers can be measured here. So the ethosomes mainly increases the drug deposition in the epidermis or the dermis depending on their composition and the drug being delivered. Stability studies. So stability is essentially for all pharmaceutical development. So the ethosomal formulations can undergo the physical and the chemical changes during storage, obviously. So water, not only ethosomes. So every formulation may undergo the physical and chemical changes during storage. So here we will validate these factors here. That is stability here. So possible changes includes the vesicle aggregation, changes in the particle size, leakage of the drug, phospholipid oxidation, ethanol evaporation, and changes in the formulation appearance are needed to be studied here. So compared to the other conventional dosage forms, here the main factor needed to remember here is the ethanol and evaporation. Okay, so therefore the stability studies should monitor the parameters such as appearance, vesicle size, PDI, zeta potential, drug content, entrapment efficiency, and the drug release here. So the formulation should be evaluated under appropriate temperatures and the humidity conditions according to the intended product and the regulatory expectations here, right? And next move on to the next advantages. So let us summarize the major advantages of ethosomes. So they can significantly enhances the drug permeation, and they also highly deformable compared with the conventional liposomes, and they can improve the delivery of the drug with the poor conventional transdermal permeations, and they can potentially provide both the local and the systemic deliveries here. So they may reduce the dosing frequency for certain drugs, and they can improve the drug decomposition in the deeper skin layers. Okay, so that's the main thing. And they can accommodate drugs with the different physiochemical properties, and importantly they provide a versatile platform for the topical and the transdermal drug delivery here. Clear? And next is the limitations. So however the ethosomes are not without limitations. So high ethanol contents may produce the skin irritation sometimes in sensitive individuals. So ethanol is volatile in nature, so changes in the ethanol concentration during the storage can alter the formulation characteristics here. So the phospholipids are susceptible to the oxidation and the hydrolysis. So the physical instability such as aggregations or the drug leakage can also occur, and the large scale manufacturing and the long term stability need careful optimization. So that's the another disadvantage. Finally, increased laboratory permeations does not automatically mean improved the clinical efficacy here. So the clinical performance must ultimately be demonstrated through the appropriate studies here. Applications. So ethosomes have been investigated for several therapeutic applications. So this includes the delivery of the anti-inflammatory drugs, antifungal agents, antiviral agents, hormones, anesthetic agents, and other therapeutic molecules here. So they have also been investigated in the cosmetic and the dermatological applications also. So for example, ethosomal formulations may be designed to improve the delivery of the active ingredients into the skin here. So their ability is to enhance the dermal deposition, make them particularly interesting for the dermatological therapy, right? Anti-inflammatory applications. So anti-inflammatory drugs are an important area of investigation here. So the objective is often to increase the drug concentration at the site of inflammation while minimizing the systemic exposure here. So an ethosomal formulation may increase the penetration into the skin and underlying tissues here. So this could potentially improves the local therapeutic activity here. So however, whether the ethosomes provides clinically meaningful advantages over the conventional topical formulations must be established through comparatively pharmacodynamic and the clinical studies, right? And antifungal applications. So ethosomes have also been studied for the antifungal drug delivery also. The fungal infections of the skin may require adequate drug concentration within the stratum, corneum, and deeper epidermal layers. So a conventional formulation may not always achieve the sufficient penetration here. So by improving the dermal decomposition, the ethosomes may increase the amount of the drug reaching the target site here. So again, the formulation optimization is drug specific, because antifungal molecules differ concentrability in solubility, lipophilicity, and the membrane interactions. And next, antiviral applications. So antiviral agents have also been explored using the ethosomal delivery here. So for topical antiviral therapy, the objective may be to achieve the higher drug concentration at infected skin or the mucosal sites. So ethosomes can potentially improve the local delivery while avoiding the some limitations of the conventional formulations here. However, the clinical benefits depends on the drug infection site, required concentration, and the biological targets. Cosmetic applications. So ethosomes can also have applications in the cosmetical research technologies. So they can be used to enhance the delivery of the certain cosmetic actives into the skin here. So the potential application includes the delivery of the antioxidants, vitamins, and other bioactive compounds here. So the major attraction is controlled and enhanced deposition within the skin. So however, the cosmetic formulations must also consider the sensory properties, irritations, stability, regulatory requirements, and consumer acceptability also. Ethosomes versus liposomes. So now let's compare both the ethosomes with the conventional liposomes here. So both systems are the phospholipid based vesicles only. So however, the conventional liposomes generally contains a little or no ethanol, okay? So whereas the ethosomes contains a relatively high concentration of ethanol, okay? So this gives the ethosomes greater flexibility here. So because of the ethanol only, the ethosomes are highly flexible in nature. So the liposomes are excellent versatile drug carriers here, but their penetration to the intact skin is often limited here. So ethosomes were specifically developed to overcome some of these limitations, okay? So therefore, for transdermal delivery, ethosomes may offer advantages over the conventional liposomes here, right? And ethosomes versus transfersomes. So the transdermals are another important ultra deformable vesicle systems here. So the transdermals are typically contains the phospholipids and an edge activator such as suitable surfactants here. So the edge activator mainly increases the membrane flexibility here. So the ethosomes intact rely sustainability on ethanol mediated modifications of the vesicle and the skin here. So therefore the ethosomes edge activators drive in deformability. So the ethosomes ethanol drive and flexibility and the skin interactions. So both systems can enhance the transdermal delivery, but their mechanisms and the formulation composition are different here. Okay, so that's the main difference. And ethosomes versus transethosomes. So now let us see about these differences also. So the transethosomes mainly combines the characteristics of the ethosomes and the highly deformable vesicle systems here. So they generally contains the ethanol and an additional edge activator. So here we are using both the edge activator and ethanol. Okay, so therefore the transethosomes can have enhanced deformability compared with the classical ethosomes, right? So why? Because means here we are using both the ethanol and the surfactants here. So that's why these are more effective compared to the ethosomes. So these can potentially improve the penetration. So however, the addition of more components also increase the formulation complexity here. So therefore the transethosome should not be automatically be considered superior. Okay, so their performance must be demonstrated experimentally for each drug and applications here. So let's summarize the comparison. So the conventional liposomes are the phospholipid vesicles with relatively limited skin penetration. Transdermals contains an edge activator and a high deformability. Ethosomes contains a surfactant ethanol and exhibits enhanced flexibility and the skin interactions here. Transethosomes combines both ethanol with an edge activator. So thus the key formulation difference lies in how deformability and the skin penetration are achieved here. Factors affecting skin permeability. So many factors affects the ethosomal skin permeation here. So the formulation factors includes the ethanol concentration, phospholipid concentration, vesicle size, drug loading and the surface characteristics here. So the drug factors includes the molecular size, lipophilicity, ionization and the solubility. So the biological factors includes the skin thickness, hydration, age, anatomic site and the skin conditions. So the experimental factors includes the temperature, oscillations, application area, dosage and the duration. So therefore comparing two ethosomal formulation requires the strict control of all these experimental conditions here. Optimization using QBD. So modern formulation development increasingly use this QBD. What is QBD means? Quality by design. So instead of changing one variability at a time, we identify the critical material attributes and the critical process parameters here. So for ethosomes, the potential critical material attributes includes the ethanol concentration and the phospholipid concentration and the drug concentration. Okay, potential critical quality attributes includes the particle size, PDI, entrapment efficiency, deformability, drug release and the permeation. So design of the experiments can then be used to understand the interactions among the formulation variabilities here. So this approach can produce a more robust formulation than empirical trial and error development. Okay. So moving from laboratory scale to the industrial scale presents some additional challenges here. So mixing conditions that work at the small scale may not produce identical results at the large scale. So shear forces, mixing efficiency, temperature control, ethanol environment and processing time can all these factors can change. So the manufacturing process must therefore be reproducible here. So in addition, the industrial development must consider the raw material quality, microbial control, packing compatibility, stability, and the regulatory requirements. So therefore the successful laboratory preparation is only the first stage of the pharmaceutical product development here. Okay, packing and the considerations. So packing is particularly important because ethanol is volatile in nature. So a formulation stored in an unsuitable container may experience the ethanol loss here. So this can alter the vesicle characteristics and the potentially reduces the performance here. So therefore the container should provide the appropriate protecting environment against evaporation and the environmental exposures here. So the packing system should also be compatible with the phospholipids and the formulation components here. Container closure integrity and the stability testing becomes important during the product development. Okay, clinical translation. Clinical translation. So one of the important point for the MPharm students is the difference between the promising laboratory results and the clinical success. So many ethosomal formulations demonstrate enhanced permeation in vitro or ex vivo. So however the clinical translation requires evidence of the safety, efficacy, reproducibility, patient acceptability, and the long-term stability here. So an increase in the flux in a frange diffusion cell does not automatically mean the increased therapeutic benefits in the humans. So therefore the formulation science must ultimately be connected with the pharmacokinetics, pharmacodynamics, toxicology, and the clinical outcomes here. Are you understanding? Yes. Next, recent research direction. So current research is moving towards more sophisticated ethosomal systems here. So the researchers are investigating the hybrid vesicles, transethosomes, stimuli responsive systems, targeted delivery, combination therapy, and incorporation into the advanced topical doses forms here. So nanotechnology and QBD approaches are also being integrated into the ethosomal developments here. So another important direction is improving the stability and the translating laboratory scale formulations into commercial viable products. So thus ethosomes remains an active area of the research within advanced transdermal drug delivery. Key points for examination here. So for examination purpose, remember these key points here. First is ethosomes are the ethanol containing phospholipid vesicles. So that is the first point needed to be remember here. And second is the ethanol increases the vesicle flexibility and modifies the skin barrier. Third, the main objective is enhance the transdermal and the dermal drug delivery. Okay. And fourth, major components are the phospholipids, ethanol, water, and drug. So all these are the main important components of the system here. Fifth, important preparation approaches includes the cold and the hot methods. So we already discussed all those. And sixth, important evaluation parameters includes the vesicle size, PDI, Gita potential, entrapment efficiency, deformability, drug release, and the skin permeations. Sixth, comparison between the conventional liposomes, ethosomes generally provide the improved skin penetration because of their ethanol mediated flexibility and the interaction with the skin lipids here. So let's quickly revise some potential viva questions here. So question 1, what is the major distinguishing compartment or component of the ethosomes? So the answer is ethanol. Question 2, so why is the ethanol incorporated here? So in order to increase the vesicle flexibility and modify the stratum corneum lipid structure here. Question 3, so what is the major barrier to the transdermal delivery? So the answer is the stratum corneum. 4, so which parameter indicates the vesicle deformability? So a suitable deformability or the elasticity test can be used to quantify the ability of the vesicle to passes through the defined membrane pores here. 5, name another ultra deformable vascular system, that is transversomes. So what is the major difference So transdermals mainly use the edge activators for the deformability, whereas the ethosomes rely heavily on ethanol. So let's revise the entire topic in 1 minute here. So ethosomes are the flexible phospholipid vesicles containing a relatively high concentration of ethanol. So the main purpose is to improve the topical and the transdermal drug delivery here, and the phospholipids from the vesicular membrane. So ethanol increases the more flexibility and the interacts with the stratum corneum here. So the flexible vesicles can deform and move through the narrow pathways. So the formulation can improve both drug deposition and the permeation here. So the important permeation methods includes the cold and the hot methods. So important evaluation parameters includes particle size, PDI, zeta potential, entropent efficiency, morphology, deformability, drag release, skin deposition, and permeations. Okay, and the major advantages are enhanced skin delivery and the flexibility here. So the major limitations includes the possible irritations, ethanol volatility, and the lipid instability, and the challenges in scale-ups. Okay, so that is the complete conceptual framework of the ethosomes here. So that is the complete conceptual framework of the ethosomes here. Conclusion. To conclude, ethosomes represents an important advancements in the vascular drug delivery. So their significance comes from the unique combination of the phospholipid vesicles and the ethanol. So ethanol modifies both the vesicle membrane and the skin barrier here, so allowing the formulation to achieve enhanced deformability and improves the drug transport here. So however, the successful ethosomal formulation requires a careful optimization of the ethanol concentration, phospholipid concentration, vesicle size, drug loading, deformability, stability, and the skin permeation here. So for an MPharm students, the most important concept to remember is this. So ethosomes are not only the liposomes containing the ethanol, so they are engineered flexible vesicular carriers in which the ethanol plays a crucial and a critical role in membrane modifications, skin interactions, and enhanced transdermal delivery. So understanding this principle makes it much easier to understand their formulation, mechanism, evaluation, advantages, and applications. So thank you for watching this lecture. So this is Madikwaris. So keep learning, keep exploring pharmaceutical sciences, and keep connecting formulation sciences with real therapeutic applications. Thank you once again.